Assalamu alaikum. I'm Suma Ahmed Junior, first year student of Abdul Malik Medical College, Noakhali. Today our topic is maturation of ovarian follicles. The following changes are observed in stages of maturation of ovarian follicles: primary follicles, secondary follicles, tertiary follicle, ovulation, corpus luteum. Primary follicles. Single layer of latent follicular cells of primordial follicles become cuboidal. Thereafter, the follicular cells proliferate by mitosis to form a multilayered mass of primary follicle. The primary oocyte with eccentric and enlarged nucleus increases in cytoplasmic volume and reaches a diameter of 125 to 140 micrometer. The plasma membrane of the oocyte is also known as the vitelline membrane. Patches of proteoglycan-rich materials are deposited between the inner layer of follicular cells and the oocyte. Subsequent fusion of these materials forms a thick striated membrane, the zona pellucida. Both the oocyte and follicular cells contribute to the formation of zona pellucida, and the striations of the membrane are caused by interdigitations of microvilli from both the participant cells for exchange of nutrient and other metabolites. A thick basal lamina appears outside the mass of follicular cells and marks the boundary between the avascular follicle and the surrounding stroma. The stroma cells of the ovary assemble around each follicle to form the theca folliculi, which subsequently differentiate into inner cellular and vascular theca interna and outer fibrous theca externa. The cells of theca interna possess ultrastructural characteristics with numerous mitochondria, smooth endoplasmic reticulum, and lipid droplets, which are essential for synthesis of steroid hormones. Secondary follicles. As the follicles grow due to increase in the size and number of follicular cells, numerous fluid-filled spaces appear between the follicular cells. All these spaces coalesce to form a single cavity known as the antrum folliculi, which is filled with liquid folliculi. This stage of development is known as secondary follicles. The liquid folliculi contains hyaluron, growth factors, several steroid-binding proteins, and some steroid hormones: progesterone, androgen, estrogens secreted by follicular cells. The antrum folliculi separates the follicular cells into an outer layer, the membrana granulosa, and an inner layer, the cumulus ovaricus, which surrounds the oocyte outside the zona pellucida. Both layers are continuous at one pole of the secondary follicle. The follicle is now about 200 micrometer in diameter. Tertiary follicle. As noted earlier, out of 5 to 12 follicles undergoing maturation in each menstrual cycle, only one follicle from the two ovaries matures fully and reaches the tertiary stage. Rest of the follicles become atretic. Such mature follicle is known as a Graafian follicle, which is about 10 mm in diameter. Selection of a single chosen follicle for full maturity amongst many secondary follicles remains enigmatic. Ovulation. It is a process of rupture of the Graafian follicle and liberation in the peritoneal cavity of a cellular mass containing the secondary oocyte and first polar body within the zona pellucida, which is surrounded by the radiating cells of cumulus ovaricus, the corona radiata. The entire mass is immediately picked up by the fimbriated end of uterine tube. Ovulation takes place in approximately the middle of the menstrual cycle, around the 14th day of 28-day cycle. The rupture is believed to result from the action of hydrolytic enzymes on the surface of the follicle, possibly under the control of prostaglandins. Corpus luteum. After ovulation, the wall of the follicle collapses and it becomes folded. The basal lamina between the theca interna and granulosa cells disintegrates and blood clot appear in the interior of collapsed follicle. The cells of membrana granulosa increase in cell size, not in number. contain yellowish carotenoid pigment and are converted into granulosa lutein cells. The solid mass thus formed is known as the corpus luteum. The blood vessels grow from the periphery in centripetal direction. The cells of thicker interna enter the corpus luteum as smaller, thicker lutein cells. About 80% of the parenchyma of corpus luteum is derived from granulosa lutein cells. Thanks a lot for your attention.